right, now let's talk about section 2 in chapter 14, specific human genetic disorders. Um, so, when you get mutations, you this can result in a change in the genotype, which can result in a change in the phenotype. For example, if there is a change from this genotype to this genotype, the double recessive from the heterozygote, if this recessive allele causes some kind of disease, then that's going to result in a change in the phenotype. Now, a change like this, from the homozygous dominant to the heterozygote, will not necessarily have a change in the phenotype. It's a mutation, but it doesn't necessarily result in a different phenotype. Of course, when the mutation alters the phenotype, that you might say it's more significant. Um, Now, when do these mutations occur? Well, if you have, let's say, two parents, okay, and so they would pass on their specific alleles, their specific genes to their offspring, if during the formation of gametes, during meiosis, a mistake is made and this allele mutates to the little a allele. Now, this does not affect the apparent parents because it's just something that's happening in their reproductive organs where they're making gametes. If this mutation occurs, then even though their genotype really is big A, what they are passing on to their children can be different. So now, even though we have parents that between them, they shouldn't have any kids with this disease, if the mutation occurs, then they can have a kid with this disease. That's not unheard of happening. Um, Now, typically, people that's not often the case. Typically, people are just carrying these mutant alleles, these mutant genes around, and so there's always a chance they'll pass them on to their kids. Here's one example, sickle cell disease. So hemoglobin is a protein that's important for development of red blood cells, and the hemoglobin not only forms part of the structure of the cell, but helps with carrying oxygen. And so there's a particular gene that's responsible for helping to make essentially the protein, one of the proteins that becomes hemoglobin. Hemoglobin consists of 4 proteins, but of course they all 4 have to be in good shape for it to function properly. So here you can see is the normal hemoglobin DNA, and there's a specific sequence here, CTT, in a specific spot, and it leads to GAA, and that codon codes for glutamic acid. But if you get a mutation, here a switch to CAT, which now changes your codon, which changes your amino acid, that single change, that single point mutation, resulting in just one codon being off and one amino acid being off in the hemoglobin protein, has a significant effect on the phenotype. And the disease gets its name from the fact that the mutation causes the red blood cells to get this sort of arched or sickle shape, and they don't flow through the blood vessels quite as smooth. They tend to get sort of clustered and block the veins and arteries somewhat, and this has all sorts of ramifications for that individual. Um, Now, so if we say the big S is the normal allele and the little s is the recessive one that causes the disease, of course there's 3 different genotypes possible. These individuals are normal, they don't have sickle cell anemia. These individuals have sickle cell disease, and these individuals, the heterozygotes, um, are typically normal, although they can have sort of mild forms of the disease. Um, Now, sickle cell is interesting in that it's more prominent in certain populations, in particular in people of Central African descent, which is a lot of African Americans. And in fact, I think I saw a statistic that something like 1 in 12 African Americans are carriers of this trait. Um, so the allele is fairly frequent. And so the question is, why is that? Well, what happens is in so these little s little individuals have the disease. These guys don't. The heterozygotes are probably the most interesting in that in regions where you have high rates of malaria, individuals with the little s allele are at an advantage. It gives them a certain level of resistance to malaria. So these individuals will get malaria. These individuals won't get malaria, but they have sickle cell disease. These individuals are resistant to malaria largely, and they don't really get that sick from the sickle cell allele. And so there's strong selection or favoring of these heterozygous individuals, which means that this little s allele is very common in these areas where you have malaria. Um, so there's an advantage to having the allele. You don't want to have two copies of it, but having one is advantageous in these particular areas, again, where malaria is common. Um, Here's an example, and this is one that's more common in people of European descent, which points out that different ethnic groups, different populations, because of where their origins are, they tend to carry different types of mutations for different types of diseases. Um, so cystic fibrosis, as we see here, is a mutation that occurs on chromosome 7. Again, it changes a specific amino acid in a specific gene that's responsible for a particular protein. And this protein forms a membrane protein that's important with transport of ions across the membrane. And when you have the defective form of it, um, that is, you have two copies of that, of this recessive, then you'll have this disease, cystic fibrosis, which is a problem in your respiratory system, your lungs, in that you produce mucus in your lungs, and you have little hairs in your lung that help move it around and get it out of your lungs. And the mucus sort of protects your lungs to a certain extent and allows for the removal of little dust and pollen and whatever that you breathe in. It helps get it out of there. Well, what happens with people with cystic fibrosis is they produce an abundance of this mucus and they can't get it all out. Um, and so there's different treatments for this, but it's a rather unpleasant condition, and it can ultimately lead to early mortality. Um, Now, it's thought that, so we'll just label it F, big F, big F, big F, little f, little f, little f. So these individuals don't have cystic fibrosis, these do. And what counts for this disease being relatively prominent, in particular in European populations? Well, individuals who have one copy of this allele tend to be more resistant to a disease called typhoid, whereas these individuals, the big F, are more susceptible to typhoid. So like cystic fibrosis, being the heterozygote has a certain advantage, but because of that, that means there's a certain chance that these two individuals, whoops, not an F, E, but an F, are going to get together, and so 1/4 of their kids will have this disease. And so whenever an allele in the heterozygous state confers a certain advantage, that allele will tend to be at a higher proportion than otherwise you would find in that population. All right, Huntington's disease. This is an interesting one because it's due to a mutation of an allele that is the dominant form. So most individuals, here let's look at this situation here. Most individuals are little d, little d. They do not have Huntington's disease. But again, if a mutation occurs during meiosis, then a child can sometimes inherit that mutation and they will have Huntington's disease, and then they can pass it down to their offspring. And so you can see if you have a parent with Huntington's disease, half the kids on average are going to have the disease, or there's a 50% chance for any kid that they will have the disease. This is a mutation that occurs on chromosome 16, and there's a portion of this chromosome where you have a bunch of these CAGs just repeated again, CAG, CAG, and having these repeats in this section of this chromosome is normal, but you tend to have relatively few of them. When mutations cause an increase in the number of these repeats, that's when individuals are affected. You see carriers have an intermediate amount, but those individuals that are affected have a really high amount. And so essentially, these individuals all have Huntington's disease, but to varying degrees. So essentially, if you were this genotype, you'd have a really bad case of the disease. These guys have the disease, which is still not a good situation. And of course, these guys do not have the disease at all. Okay. Now, what's it going to take for an individual to have this genotype? Well, it's going to require that both parents have this allele and have Huntington's disease. Oops, big D, big D. That's the only way you will get a child that's homozygous. And the allele is infrequent enough that that's not terribly likely that two parents who both have this disease are going to get together. Huntington's disease is also interesting because it's something that affects people at middle age, so they often don't know they have it until after they've had kids. Now, if it is in a family, that is, it's out there, then you'll know that there's a chance you may have inherited it. And so people who have a history of Huntington's disease in their family will often get themselves tested to see whether or not they have the disease, whether they have that big D allele. All right, so we've talked a little bit about already the advantages of these mutant alleles. Again, in certain situation, those heterozygotes can have an advantage over the homozygous dominant individuals because that recessive allele gives them a certain resistance to the disease. And they also have an advantage over the homozygous recessives because they have the disease. So that's what keeps these, in certain situations, that's what keeps these recessive alleles present in the population. The fact that they can be found in heterozygous form, and also if those heterozygotes have an advantage. All right, so we can also get larger chromosomal disorders, and in particular, again, during meiosis, sometimes things don't go correctly, and that you get a phenomenon what's called non-disjunction. So as you can see here, normally in this situation, the red pair is a homologous pair, the yellow pair is a homologous pair. You should produce two cells that are like this. That is, come on, they have one red, that's the red one, and one yellow. But sometimes the chromosomes don't separate correctly during meiosis. So you can see in this situation, they each have one red, but this one has two yellows, and this one is missing a yellow. So now we've got this problem here, because then during meiosis 2, you can see they separate properly, but now we have gametes that should only, in this simple situation, have two chromosomes, but they've got three, and we've got gametes that only have one chromosome. So some have an extra chromosome, and some are missing a chromosome. And I'm running out of time, so in the next video I will explain how that's a problem.